quick, Sandra. So today is a shared seminar between the Prepare Institute and the Human Milk Institute. And this project started a little bit over a year ago, is that right, Sandra? I see Rodnish on the call there as well. As a seed project that both HMI and the Prepare Institute co-funded with the idea to fund two investigators that probably hadn't worked together and come from two different disciplines to specifically address the need to put more resources and more tools behind screening the safety of breastfeeding and human milk during pandemics or other looming emergencies. And that's when we got a wonderful application from Sandra and Rodnish that they're going to present the results today. Sandra is an associate professor in the Department of Pediatrics, same division as I am in neonatology. So we have a neonatologist and then someone who has nothing to do with babies, really, Rodnish, who's more the mastermind behind the technology that Sandra's going to present as well. All right, Sandra, all yours.
I'll share my screen. Let me just exit this floating. Okay. Perfect. Can you see my presentation? Thumbs up. Yep. Excellent. Okay. So thank you, Lars, for inviting me to speak about the project that I was able to work with Retnesh, which, again, I had to really learn a lot of bioengineering terms and what everything meant and, you know, how kind of go back to my physics roots on, electrical conduction, et cetera. So hopefully I'll be able to break that down to a bunch of biologists that are listening in. But first, I just want to kind of show an introduction for using breast milk as a kind of detection system from a pandemic standpoint. And then I'll finish off with the discussion about this device. So the objectives of my talk today is to outline the impact of pandemics on breastfeeding and human milk, to describe the pathogens that can actually cross into human milk, and then describe the bioactive molecules in human milk that can actually combat those pathogens in order to not induce infection in the actual newborn or infant that is breastfeeding or ingesting the human milk. And then finally introduce the GFET detection of cytokines and viruses in human milk samples, which is the project that I worked with in Retnesh, and I'll describe what GFET is, etc. Okay, so let's talk about breastfeeding during a pandemic. Here's just a quick little picture of what a mammary gland looks like. You could see it looks like a bunch of grapes or alveoli, which is interesting because I study the lungs. And you could see here that in the alveolar part of the milk duct, you see these epithelial cells that lipid droplets kind of cross, and then they make milk secreted into the actual alveolus, and then it goes down into the duct, into the mouth of the baby. Okay, so we'll start with, I'm going to talk about two recent pandemics. The first one is the HIV pandemic. This began in the early 1980s with the first recognized cases of what would later be called AIDS, and that was reported in the US. It was first detected due to unusual clusters of infections, including Kaposi sarcoma and pneumocystis pneumoniae, which was initially observed in young gay men. And again, this I'm bringing up because this kind of prevented the quick detection and the quick thinking of maybe checking other bodily fluids, including human milk. And so I think we've learned a lot from the HIV pandemic of what to do and what not to do. Researchers identified the causative agent of AIDS as human immunodeficiency virus, or HIV. And then by the mid-1980s, it became clear that HIV was not confined to just a single group, such as young gay men, or a single region, such as the US, either Los Angeles or New York, but it actually expanded worldwide and in all human populations. And so the HIV pandemic, mother-to-child transmission was recognized. Again, a little probably later than we would hope, but it was something that was seen as a significant route of infection. And here I'm just showing a little picture of an HIV positive mom and the various routes of mother-to-child transmission. Most of the cases were attributed to transmission during pregnancy or delivery, which you see here in these first two boxes. Although what the epidemiologists and clinicians actually then saw is that some infants that were born to HIV positive mothers who had then after delivery tested negative for HIV were then later tested positive, which suggested a postnatal transmission. And that's when this possible transmission via breastfeeding was hypothesized. So then epidemiological evidence showed a correlation between breastfeeding by HIV-positive mothers and subsequent infections in children. And this was noted mainly in African regions that had extremely high rates of breastfeeding. And that actually showed that large population of postnatal HIV-positive newborns and infants. So then to confirm the epidemiological findings, clinical studies were then conducted in the 1990s in Africa, which provided a definitive evidence of breastfeeding as a route of HIV transmission. Specifically, randomized control trials were actually utilized to test antiretroviral therapies, or ART, in breastfeeding mothers, and they actually found that those ART therapies actually reduced HIV transmission to their infants postnatally. So that was kind of the first clinical trials providing evidence that it was transmitted through breastfeeding. And then studies comparing both breastfed and formula-fed infants born to HIV-positive mothers did show a higher rate of HIV infection among the breastfed group. And then transmission rates at that time ranged from 14 to 40%. So breastfeeding was still recommended in, again, the areas where it is very easy to drink infected water. And so in those areas where transmission rates, aren't 100% or even 80%. So because, there was such a large range and transmission rates weren't actually that high, even though they could be as high as 40%, in those areas that, drinking polluted water or infected water is a more a bad way of affecting infants, then breastfeeding was actually still recommended in order to protect infants from infectious diseases found in their water supply. Public health guidelines in more water-safe areas began emphasizing formula feeding as a safer alternative for HIV-positive mothers, obviously where feasible. And then with that advent of the antiretroviral therapies, or ART, it dramatically reduced the risk of transmission during breastfeeding to less than 1%, which was a huge win for the breastfeeding community. So HIV in breast milk. And the way that it was detected was through various molecular mechanisms, including assessing RNA, DNA, as well as the P25 antigen. And then the mechanism of infection to an infant from its mother was that the virus actually crosses from the maternal bloodstream into the mammary glands, which I showed a picture of, through what's called the blood-milk barrier. And here's box 1 that actually shows that transmission of the virus through the epithelial layer into the milk duct, or the alveoli. And then the virus itself can actually form or be present in the milk in 3 different ways. It could be within a cell-associated virus, so it's actually infected an immune cell that's present in the human milk, and as the immune cells passes through into the baby, so does the virus. It could be as just a free viral form, or a vesicle associated viral form. So those are the different mechanisms that it could remain in the breast milk and then pass to the infant. And then infection mechanisms in the infants after ingestion of infected human milk included access to the tonsillar, the respiratory, and the intestinal epithelial cells. So factors influencing transmission, like I told you guys, there was actually a pretty large range of transmission, which was reduced significantly by ART. So viral load, higher viral loads in breast milk are associated with increased risk of transmission. Persistence of viral shedding, the more persistent the viral expression in milk over time, obviously the higher likelihood of transmission. The composition of breast milk. So there are many antiviral factors in breast milk or human milk. And I will be discussing those at a later time point, and they can actually affect viral infectivity. Duration and frequency of breastfeeding. We recommend exclusive breastfeeding. And so if it is exclusive, then that, especially in those moms with HIV, then prolonged and frequent breastfeeding does increase exposure and potential transmission risk. And then finally, the infant's own immune system. So the maturity and effectiveness of the infant's immune response, especially in the gut, which is one of the main targets of the different viruses in human milk, they play a crucial role in determining whether exposure leads to infection. And as a neonatologist, I know that, a term infant's immune system isn't, the best or the most mature, but a preterm's immune system is even less mature, putting that infant at a more higher risk of developing infections, especially through human milk. Before I start into this pandemic, you do notice that I am using human milk and breast milk interchangeably. It's usually based on the articles that I had read and the terminology that they were using. And obviously, I was using a lot of older ones from the HIV pandemic. So if you hear me just use them interchangeably, just know, they mean the same thing. Okay, so now to the most recent pandemic. Which was a COVID-19 pandemic, which began in Wuhan, China, with the identification of a novel coronavirus, SARS-CoV-2, that was linked to a seafood market at the end of 2019. Early cases presented with severe pneumonia, and the virus was quickly identified as highly transmissible. And I think this differs from the HIV pandemic because there wasn't a specific group that had these kind of like weird symptoms, like the rare cancers that they noted. This was already seen in multiple populations and had the respiratory symptoms that were not, didn't take a long time to develop like Kaposi sarcoma does. So the World Health Organization declared COVID-19 a global pandemic on March 11th, only a couple of months after the first cases appeared in China, as cases surged worldwide, very, very highly transmissible. And then rapid human-to-human transmission led to widespread lockdowns, travel restrictions, and overwhelmed healthcare systems. And I'm going to talk about how those affected breastfeeding as well, not only just the virus itself. So COVID-19 pandemic had some negative impacts of breastfeeding, and these included whether SARS-CoV-2 could be transmitted through the breast milk, which led to a lot of mixed recommendations from a lot of like the CDC and the WHO. And even individual healthcare providers advised temporary separation of COVID-19 positive mothers from their infants, which again discouraged breastfeeding and increased the use of formula. And anyone that was, in Jacobs or any kind of delivery center during that time knew that there was always, new recommendations based on whatever data was coming in, because obviously I just told you that an infant's immune system isn't very robust. And so if, this virus was very deadly to, adults, as we had, finally figured out, we did not want to have those infants at risk. And so there were initially lots of separation guidelines in newborn infants along with their mothers. And then even when they were starting to be allowed to room in together, then the fathers were discouraged to be part of the whole birth process. So this was very, very difficult for a lot of new families during this time. And then finally, lockdowns and healthcare system strain reduced access to lactation consultants, peer support groups, as well as maternity care services. And again, we saw this as well in our center, even just being able to be part of deliveries as the neonatology group, we had to have like a dirty team and a clean team, which just meant who's going to be doffing all of the PPE and being in the room there, accessing the actual delivery, being able to help with the delivery. And then if that baby for some reason needed to actually be then transferred to the NICU for further care, then that baby had to be passed on to the team that isn't doffed. And so there was a lot of healthcare system strain, and that was just from the standpoint of just the neonatal group. So I can even imagine all the other groups that were strained as well. And then finally, the mothers themselves. I kind of told you all about the different advisories and guidelines that were provided in the hospital. So you could even think not only does the medical team strained, but the pandemic heightened stress and anxiety among mothers, which, as we all know, stress is not a good way to increase milk supply, and as well as confidence in their breastfeeding. But there were also some positive impacts on breastfeeding during the COVID-19 pandemic. So this is Tina Chambers' paper that I'm quoting. They actually, and I know Lars, you were involved with this as well, they actually looked to see not only whether there was SARS-CoV-2 RNA detected in the human milk, but whether it was replication competent virus. And this was a huge point of discussion because just because there's RNA, that doesn't mean that the virus is actually able to replicate and then infect, because just bits of RNA will not infect a child that is ingesting that breast milk. And so in a positive way, because replication competent virus was not detected in human milk in mothers that did have SARS-CoV-2 RNA, the risk of transmission via breast milk was not likely. And that was the exact words that was written in that paper. So that was great news. And then breastfeeding was also found to provide protective antibodies, including IgA, which you see here as this little purple double-ended, like two Ys put together, against SARS-CoV-2. And then when vaccinations were then implemented in 2021, then vaccinated mothers were shown to even have more of these immunoglobulins, which then could protect their infants, which were unable to obviously obtain the a vaccine. And then by that point, we already knew kind of the demographics of the virus, and that the younger children really, even though they would be infected, they really weren't as symptomatic as maybe older children or children with comorbidities, etc. So that was also something that compounded with, all these positive features of human milk. And then for some mothers, as I said, the stress of the pandemic was difficult to overcome. But for some other mothers, they couldn't go to work anymore. And so they either stayed home or did their work remotely, which actually created an environment that's more conducive to breastfeeding, because then they didn't have to go to work and just pump, which again makes milk production difficult and then reduces time for bonding and breastfeeding. So that was very positive, and definitely saw a reduction in maternal stress, depending on which side you were on. Okay, so you could see two different pandemics, two different responses, two different timelines. The HIV pandemic really relied on epidemiologic data, looking at the populations that were breastfeeding more than formula feeding. Lots of guidelines based on that the HIV virus was replication competent in the human milk and therefore did have a risk of infecting the infant, versus SARS-CoV-2 that was not replication competent, therefore reduced like zero ability to infect an infant, at least through the human milk. And you can see the different timelines, how quickly we responded to the SARS-CoV-2 pandemic, again, because it was probably more highly transmissible and affected everyone, versus the AIDS pandemic, which initially infected a group, which was a minority group that the government really didn't seem to want to invest their time in initially until other groups became infected. So now let's talk about how viruses actually access human milk, and which ones are the ones that are replication competent and should be monitored for, and why we want to use a detection system in human milk. So viral pathogens, we know that breastfeeding is extremely beneficial on both short and long-term health outcomes of the mother and infant. I see a lot of human milk investigators and lovers here on the presentation, so they understand. But human milk also, not only does it have these benefits, but it also protects against infections in the term as well as the preterm newborns. You could see here this figure from this paper where human milk has a variety of pathogens that have been detected. Here, there's 25 human pathogens that have been detected in human milk. And then when you're actually looking to see whether they're able to replicate in human milk, which means that they are, can be infectious in the newborn, you can see that there's only 10 human pathogens that are replication competent and can infect the newborn baby. So these are the ones that we really, try to avoid breastfeeding in if possible. So kind of what I already said, even though they're detected in the human milk, they rarely lead to infection or disease. And then one of the thoughts for even the replication competent viruses is they don't cause infection or disease in infants because there's a lot of bioactive components of human milk. And again, I'll talk about those coming up. And then I really like this figure because this kind of shows the difference in 4 viruses that are replication competent that are found in human milk, and kind of what the guidelines are for breastfeeding. So here we just kind of look at transmission rate. This is HIV, human CMV, Ebola virus, and T lymphocyte virus. And then the current recommendations. And so again, this is split up into high income settings and resource limited settings. And the high income settings, they actually are recommending to not breastfeed regardless of ART and maternal viral load, but hopefully that is changing. Henry Lee just sent out a little survey to ask the neonatologist, how comfortable would you feel knowing that this mother is on ART and her viral load is undetectable? Would you be comfortable to advise her and guide her to breastfeed? And I absolutely do support that. So I'm glad that high income areas are now understanding, the actual biology of transmission. And if there's no viral load, there's no virus to transmit, and breastfeeding is important. Versus resource limited settings, there's always been a recommendation to exclusively breastfeed, even if the mother wasn't on ART, just because of that, transmission risk. But now, hopefully, ART itself, like I said, reduces that risk to less than 1%. CMV, you can see the transmission rate is a little, kind of more static, less kind of broad than HIV. And there's no recommendations against breastfeeding except for the specific patient population, which is the very premature baby. And that's kind of the only thing that we do have to think about as clinicians, except that we don't have a way to detect CMV in human milk in the NICU, especially in those mothers that are providing their breast milk to their premature babies. Ebola virus, you could see the transmission is unacceptably high. Suspend breastfeeding, WHO recommendation, absolutely not. And then HTLV, again, kind of the same risk as HIV, and the CDC recommends no breastfeeding of infants. So just kind of another really cool figure to kind of show you guys that even though these are all replication competent viruses in human milk, their recommendations are very different. So now let's talk about what's in human milk that can actually hopefully suppress actual infection, which then will lead to disease of the newborn. So here is just a nice little figure that shows various antiviral components in a drop of human milk, including HMOs, glycosaminoglycans, immune components, cholesterols, mucins, lipids, et cetera, and just some mechanisms of action, including just like killing the virus outright, inhibiting the ability of the virus to bind to various receptors, inhibiting them just to enter in a non-receptor mediated way, and then finally inhibition of intracellular replication of the virus itself. And so let's actually talk about each of these components, and I will be very brief with this. So immunoglobulins and immunological factors, including secretory IgA, which is actually the most abundant immunoglobulin, binds to viruses, forms immune complexes to combat infection. And then IgM and IgG provide systemic immunity, usually from the mother, and activate the complement system. Non-immunological components, including oligosaccharides, promote growth and beneficial gut microbiota, mimic the host cell surface receptors as decoys, which is kind of cool. And I'll show you another protein that does the same, as well as bind to viruses to prevent them from entering the cells. There's some cytokines, and I will talk about cytokines a little bit more later on in the presentation. So IL-10 is actually the most abundant and the most beneficial, and that actually modulates inflammation. It's an anti-inflammatory. It actually preps the baby gut to really try and combat any type of viral pathogen entry. And then TGF-beta is also beneficial in that it actually promotes repair of that mucosal barrier of the gut. Lactoferrin, it also actually kind of mimics the host cell surface receptor as a decoy, but also sequesters iron, which then inhibits the viral replication because they do require iron in order to make more little virus particles. Extracellular vesicles, it's a very hot topic now. Lots of people that are part of HMI are actually studying this. They are kind of like little pockets that have various stuff inside them, including mRNAs. And they function in antiviral signaling, immune cell communication, and pathogen defense. Mucins, just a bunch of mucus, kind of what you think about what comes out of your nose when you're sick. They form barriers on epithelial surfaces. Specifically, again, think about the gut, think about the respiratory surface, and think about the mouth as well. So when the baby actually drinks the milk, their mucosal barrier is, the first thing that is detected by the milk. And then finally, lipids. Free fatty acids do disrupt the viral lipid bilayers. And then there's some other antiviral components as well. And again, this is not like the end all this. There's so many more things. Like human milk is amazing for what it contains. So I won't go through these, but just know that there's other stuff that can actually act as antivirals and protect the infant from infection. And I just want to quickly show this picture because it has a nice breakdown of what the various viral and antipathogen factors are in human milk. Here are some milk fat globules with the free fatty acids, and you could see here how the free fatty acids actually kind of like disrupt that enveloped virus or their lipid envelope. You can see here, and it causes the viral particle to lose its envelope, and then it's unable to infect the baby. If you look down here into more of the carbohydrates and proteins, milk contains soluble viral receptors that again also act as a kind of decoys, where the virus thinks it's binding to a receptor, but it's actually a soluble viral receptor from the human milk and not actually linked to a cell, which again allows the virus to bind, but then not actually enter the cell. And then it's swept away by the immune system. And then lactoferrin here is just kind of mentioned. It has multiple different methodologies. And here are all the different viruses for the mechanisms of action are exposed to. So you could see it's got extracellular, intracellular, et cetera, mechanisms of action to reduce inflammation or infection. And then finally, just the immunoglobulins, specifically immunoglobulin A. Again, you can see the two Y's put up together. And you could see that here it just surrounds the virus and then kind of targets it for phagocytic elimination. And then it also neutralizes, so then it can't link to its viral receptor on the cell. So very cool what human milk contains and what it can do. So what if the human milk, we have a donor milk program. What if there is a risk of, let's say, CMV, something where, it's not really transmissible, but we kind of don't want it it is transmissible, but it's not infectious, but we don't really want it in the human milk. What are some methods that we can do? And here are just an overview of various human milk pasteurization processes. The Holden standard process is great for eliminating or denaturing the virus, but it also denatures a lot of those beneficial proteins that are found in human milk. And so these innovative processes, like very high heat, very short, temperature low, all these various types of way to inactivate the virus, but also being able to maintain the actual bioactive molecules that are found in human milk. All right, so I spoke about the viruses that can cross into human milk and the mechanisms of action on infecting a baby and how the milk tries to avoid or combat that in transmission as well as infectivity. And then a way to kind of hopefully remove or at least inactivate the viruses using heat. But how do we actually detect it? And so this kind of is moving towards the actual project that we were doing. So this I'm just showing the polymerase chain reaction in case there's no molecular biologists here. And this is just a way to replicate DNA. You kind of split it apart. You have some primers that then get copied, as you can see here in the two directions of the arrow. And then those kind of like lift off, and then those get copied and on and on and on and on until you actually get kind of like a number of copies or a copy number, which is actually what it's called. But the problem with SARS-CoV-2 is we would detect copy numbers, when we do the PCR test for like nasal swabs. But what copy number actually is specific for a virus that is transmissible and infective versus a copy number that, oh, you were previously infected, but like now, the body has taken care of it and you're actually not gonna transmit that virus to, a family member, et cetera. So this was a huge problem back in the day. Okay, so here's some other molecular methods that we use to detect viruses. So I kind of talked to you about some of the molecular methods, polymerase chain reaction, and here I've also added in how long it takes to collect, prep, and then actually get a result in. And then we have some immunological methods. ELISA is a way to detect proteins, as is the Western blot, some of the things I do in my lab. Culture-based methods, this is, again, in Tina's paper, this is how they actually did it. They had to isolate the virus in cell culture and see whether it grows and is able to like infect cells and then, generate new cells, which kind of shows that it's replication competent. Electron microscopy, and then other advanced combinatory methods like flow cytometry. So then here I just kind of added some pros and cons of all the various different methods. And you can see like what I had already talked about, the PCR, you can't really distinguish between infectious virus. Everything is pretty much costly, expensive, time consuming. Sensitivity is affected in some of these methods. et cetera, et cetera, et cetera. So are we able to develop a rapid, effective, and easy-to-use test for the presence of viruses and markers of infection in human milk? Yes, we can, and I am here to show you how. So first, I want to shout out to the team that helped with this project. So Ratnesh Lal, who's on the call today, and then his student, Michaela, and his graduate student, Tyler. Tyler really described the system to me, how it worked, why graphene is so important and such a great conductor. Michaela walked me through the whole process of prepping the milk, putting it on the chip, and then how we actually obtained a direct shift and doing all the analysis. And then my summer student, Bianca, helped with the project whenever I was on service and was able to jump in and help Michaela with all the processing whenever it was needed, because we kept getting more and more milk to really not only validate the system, but then perform some of the different analyses that I'll be sharing today. Okay, so so this graphene field effect transmitter, this is kind of the overview, and I actually have a YouTube video that I want to kind of show you guys because it's better to understand it if you kind of see it in real time. So I'll X out of my presentation to show it for you. I have all the kind of like timestamps, but that's really the way I had to learn, relearn physics again. So you take our processed milk, and not only did we look at cytokines, but we looked at the spike protein of SARS-CoV-2. And then you add the sample onto the actual GFET chip, which again, I will describe in more detail. And this chip has this graphene layer, which is a very, very thin, highly conductive layer of carbon atoms. And it actually has an aptamer, and I'll describe what an aptamer is, bound to it. And graphene is very sensitive to any changes. So the way that one YouTube video that I watched described it was picture a lake and then drop a little pebble in it, and you see that kind of like the wrinkles in the water. That's kind of how graphene works, is that any kind of like stimulation is detected, and instead of obviously the rippling of the lake, it actually generates an electrical charge. And then here you can see the protein of interest. Here again, we stated a cytokine. When it binds to the aptamer, that's kind of that rock being thrown in the lake and rippling the graphene and allowing this shift in the voltage. And you can see here, this is the baseline or the negative, which is the unbound aptamer, and then the voltage moving to a positive shift after the aptamer is bound to its protein of interest. So I think this is, let me actually X out, stop share. And then hopefully you guys will be able to hear the video as well. Oops. Okay. And let me know if you guys can hear this. So now they're going to talk about, this is a group that actually developed this biosensor using graphene for detection of a COVID-19 virus. So this was in 2020, and this group was from Korea. And this is just kind of how they developed it. So I'm just going to like show you kind of the initial what graphene does, and then I'll move on that they use not aptamers, but they actually used antibodies to actually detect. So again, let me know if there's any issues with sound or anything.
Can't see anything yet, Sandra.
Oh, maybe I should share my screen. Thank you, Lars. Yeah, I didn't share my screen. I enjoyed the video. Okay, share screen. Share. Okay, you guys see this? Okay, perfect. All right, let me get rid of this so I can drop the meeting control. Okay. Okay, so this is graphene. Okay, I'm going to skip this because this is how they made it.
There's no sound on it, Sandra, so maybe you want to talk to it.
Sure, no problem. So there they show kind of the graphene sheet. And then here, they're just showing that the graphene, and again, you guys can probably see the words. So that's just what the narrator's saying, but the graphene actually bridges, again, there's source electrodes and drain electrodes, and that's what you'll see as causing that shift in the voltage. And so, yeah, if you can't hear, just read the words. And I'll talk over it as well. So they've set it up so we can actually detect the electrical changes. So P-base is what Dr. Lal's group also uses, which is why this is also important. So they added the SARS-CoV-2 protein antibody, which antibodies are naturally derived. So it bound to that P-base to form a chemical bond. So that's the link to the actual graphene. Okay, so just want to show you that. And then let me get out of YouTube and go back to my presentation. So hopefully that kind of helped with understanding, what graphene is and how we actually generate the biological detection system. So go to slideshow from current slide. Okay. So what I showed you is how they generated that graphene layer and how they made antibodies while Dr. Lal's group actually chose to make aptamers, and I'll talk about why. Okay, so functionalization is pretty much what you guys just saw on the video. So graphene is, again, that layer, very thin, 1 atom thick, very conductible. And then P-base was added, and that's, to kind of join the biological factors onto, the actual graphene sheet. And so aptamers are single-cell DNA or single-cell nucleotides, and they link to the P-base layer that sits on top of the graphene. And then it's functionalized or made to function well by multiple P-base washes and the addition of the different aptamers. And what's nice is you can actually just purchase these aptamers. So I'll talk more about what aptamers are, but these can be generated in labs, and they can be very highly specific for that target molecule of interest, whether it's the protein of SARS-CoV-2 or a specific cytokine. And so, we decided to use aptamers of a couple of cytokines as well as the SARS-CoV-2 spike protein. And in a previous publication in PNAS that Dr. Lal's group did, they had used the aptamer of SARS-CoV-2 nucleocapsid as well as spike protein and shown that it was effective in detecting nasal swabs in patients that were positive for SARS-CoV-2. So this is just more the detection system. This is what the chip actually looks like. It's actually a little smaller than this picture. It's kind of like a postage stamp. Here's the actual GFET well, or where the where this graphene sits. And this is where we functionalize it with the P-base and then add the aptamers onto it. And here you can see there's a source and there's a drain, which kind of allows the detection of that electrical current or the voltage changes. And then the biological samples are added to a functionalized GFET platform. And what you could see here are the aptamers that are bound to the P-base, that's bound to the actual graphene. And then once we add the different proteins of interest, then you can see they bind to the analyte and then result in that shift. And in Dr. Lal's group, they look at what's called a direct shift, which is the difference between the source and drain and then the gate. He will be happy to answer all those questions if anyone has any more questions about the actual mechanisms of voltage changes. Okay, so just a quick aptamer versus antibody, because that's, again, something that I really had to learn. I'm used to using antibodies. I use antibodies in flow cytometry. I use antibodies in ELISA, and these are naturally derived proteins, or I guess immunological factors that you have to actually obtain from biological species. So in order to generate antibodies against a specific virus, you need to have an animal infected with that virus. And then their B cells, which are the cells that actually generate antibodies, those are the ones that actually start secreting the antibodies against, the protein of interest. And then those antibodies have to be collected. And obviously, they're commercially available. I buy them all the time for the different proteins of interest that I'm interested in studying. You probably heard of monoclonal antibodies that are also used. Initially, were used as a therapy against SARS-CoV-2, et cetera. So these are biological, very good for what they had been used for. And now we've actually moved on to aptamers. And so here I've just generated a table that kind of highlights the differences between an antibody and an aptamer. And so antibodies, like I said, produced by immune cells, bind to specific antigens. And then these are the single-stranded DNA or RNA or nucleotides that also bind to specific targets. They're both very highly specific. And what's nice is because the aptamers, we make them, not necessarily me, but, human beings, they can actually be adjusted and their affinity can be tweaked in order to really generate that high specificity for their target molecule. Stability, the aptamers are a lot more stable than antibodies. Production, I already told you, you need a biological system to produce antibodies, whether they're full animals or cell cultures, while the aptamers are synthesized chemically, which again is cheaper, more reproducible, and that's what I have here, and much faster to produce, longer shelf life, more consistent, because again, you don't have that batch effect that you would in any kind of like biological system, et cetera, et cetera. So I don't have to go through all these, but just know that, that is a really good system that can reproduce what antibodies used to do, but now aptamers can do in a faster, cheaper, and maybe even a better way. So here's our experimental workflow that we use to assess human milk that we got from the Tina Chambers's mommy milk and biorepository. So the milk was centrifuged, filtered, and processed, because we also wanted to compare what if we just use raw milk versus processed milk, because as I said, raw milk has a lot of those proteins and lipids and factors, and so there could be a lot of signal to noise ratio, which is something that we don't want. We don't want a false shift or a false direct shift versus if we just process the milk, have it completely quiet, and then we're actually able to add in our own antibodies, which we actually did initially. We just spiked our own antibodies to actually validate the system before we actually just used natural milk with its natural cytokine or protein content. And so you could see here, we always did 2 baselines just to make sure that we had an internal control as well as just a regular baseline control. And then we spiked the milk with our target protein. And then we did a sample reading after 10 minutes. And then the actual direct shift, we compared the sample reading to that second baseline. So our project aims, I will show next. And I just, again, want to thank the Lars and Prepare and HMI for funding this project and having, a clinician and a bioengineer work together on this really cool project. So the first aim was just milk processing. No one's done this before. We didn't know, what kind of milk we were able to use in order to reduce that signal to noise ratio. Then we wanted to kind of start with just kind of cytokine detection. What are some of the cytokines that are present in human milk that may be increased in infection? So even though the virus may not actually be transmitted into the breast milk, if a mom is infected, is that detectable? So kind of like something outside of what had been previously done, where we're just specifically looking for a viral pathogen, but is there a way to also detect that mom is ill? And then another thing that hasn't really been explored is if mom does have large quantities of pro-inflammatory cytokines in her human milk, how does that affect the newborn? Even though a virus may be not transmitted, and doesn't cause disease, or whether it does or doesn't, what actually happens if mom has, high expression pro-inflammatory cytokines? So that's something that's ripe for investigation. And then finally, obviously for this Prepare Institute, we are also interested in the viral protein detection. And so we did spike the milk with SARS-CoV-2 spike protein. So aim 1, milk processing. We generated baseline characteristics and performance of the GFET sensor of whole milk compared to centrifuge milk. We wanted to limit that off-target signaling due to milk complexity, as I already mentioned. And then whole milk, which we didn't process at all, maintained all of the different bioactive factors, did result in an extremely large, so that's right here, this dark blue, in extremely large direct shift of 500 millivolts compared to filtered milk, which is not ideal for detection, especially if you want to be really specific to the target protein of interest. While centrifugation and filtration of milk resulted in a less than 40 millivolt direct shift, and 40 millivolts and above is what we had delineated as a significant change or statistically significant change. All right, then we'll talk about why we chose the cytokines that we chose for AIM-2. So we wanted to detect pro-inflammatory cytokines that are found in human milk and that may be increased in if mom is either infected or she has a kind of an inflammatory systemic state. And so there's two very effective pro-inflammatory cytokines that are found in the human milk that are absent in the newborn. And those are interferon gamma, which is here, and TNF alpha, which is here. And actually the newborn really lacks a lot of cytokines. And so the cytokines that they require, not only pro-inflammatory, but also anti-inflammatory cytokines, they receive through the milk. And so, again, we first wanted to look at the harmful cytokines, which may be, again, important in infant disease status as well, or maybe, you know, it affects the intestinal epithelial cells for some reason or another. And so interferon gamma secreted by T cells and NK cells, and it can actually increase the intestinal epithelial barrier permeability, which again is bad because it means that those cells are not sitting tight against each other. They kind of get loosened up, and that allows pathogens or maybe even bad microbes to kind of cross through into their blood system and their immune system. And then TNF alpha is also a pyrogen and contributes to systemic inflammation as well in a negative way. And then IL-6 has, you know, can be both pro and anti-inflammatory. This is actually a really cool cytokine in human milk. From the standpoint of SARS-CoV-2, IL-6 was the whole cytokine storm cytokine. So if it's generated at too high a concentration, then it can actually have a lot of damaging effects. It can cause lung damage, endothelial damage, etc., kidney damage. So IL-6 at very high levels is very bad. But in milk, it's actually found in really large concentrations in colostrum. And we know that colostrum is very beneficial to the breastfeeding infant. And so that kind of like balance of just enough IL-6 versus too much IL-6 is something, again, that would be very interesting to investigate using human milk and this GFET sensor. And so elevated IL-6 in response to maternal inflammation or infection actually may benefit the infant by enhancing immune responses. But you don't want it to overactivate the infant's immune system as well, and obviously causing that, again, cytokine storm, which we heard so much about in SARS-CoV-2. So this is our results for when we generated the interferon gamma aptamer, functionalized it to the GFET biosensor, and then performed various concentrations. And you can see here's just milk as a baseline without any spiking of the cytokine. And then 300 picomoles, 100 nanomoles, and then 300 nanomoles, you can see a nice direct shift at 300 nanomoles, which is still a super tiny amount. So very little of that protein is needed in order to generate a signal. TNF alpha showed the same thing. At 30 nanomoles, we saw a nice increase in the direct shift. And then IL-6 saw the same thing. It's not written here, but the dose was about 30 nanomoles to actually detect IL-6 that was spiked into the human milk. And then finally, AIM-3. Okay, so we validated our system. We know how to process the milk in order to reduce that signal to noise ratio, but now can we actually detect that viral protein? And so we elected to use the spike protein initially because we could do that in a BSL-2 lab. And then these are kind of backwards, usually controls on this side, but here control is orange. And here we have at 50 nanomoles of the S protein, we see a direct shift that increases from the milk control. So the protein detection is possible, at least in spiked milk. And so with that validation of we're able to spike the different proteins in processed milk and detect it using the GFET biosensor, but now can we do it in milk that came from a mom that was infected with SARS-CoV-2? Like I said, SARS-CoV-2 RNA is detected in the actual human milk, But it's not infectious and doesn't transmit and is not replication competent. So we are just interested in looking at, without having to spike the milk itself, we just processed it enough to remove the fats, but still maintained the kind of lactoserum, as some papers call it, that contains all the proteins. So we do have a lot more noise now. It's not as clean as we had when we were spiking it. But is that okay? Is the noise okay? Are we still able to detect the little bits of SARS-CoV-2 that are present in there? And we got 2 donors, again, from the biorepository of Tina Chambers' group. And we found that here's donor 1. Here you can see that the orange is the actual, so this is just how we processed it. Baseline 0, we added 10 microliters of milk. Baseline 1, another 10. And then we got our samples. So you can, and then this is how we kind of calculated them. And orange is the DS initial. So it's sample minus the baseline without any milk added. So that's where we expect the largest increase. And you could see that we did have a very large increase in donor 1. Actually, the direct shift was almost 800 millivolts. And we had said previously that 40 is, the statistically significant, what we want to see, while donor 2 had a direct shift over 60. So you could see there's a lot of batch effect, and every mother is going to have a different concentration of either virus or protein or whatever the target molecule is. And obviously, the most ideal control would be the same mother who donated the milk, but without SARS-CoV-2. But unfortunately, that sample was unavailable, but we would have loved to do that. So in conclusion, we saw significant direct shifts in interferon gamma spike human milk at the concentration written TNF-alpha, IL-6, and SARS-CoV-2. So this is primed to now be used in actual donor human milk samples and validated with ELISA-based assays. And so an ELISA-based assay is, again, using the antibody instead of the aptamer. And so we do that a lot in my lab. And so we just want to make sure that the ELISA also shows an increase in the level of, let's say, either SARS-CoV-2 spike protein or the different cytokines, and in parallel kind of show that, the direct shifts are also increased. And then that way, we validate our findings with an assay that's already been in use for dozens of years. We are still interested in determining the direct shifts of donor milk from mothers that were positive for SARS-CoV-2. And again, it'd be great if we had controls that were the same mothers, because as I showed you, the human milk is really from the mother, and its composition is really mother dependent. And so if we are able to collect from mother and then after infection, that would be ideal. We are also interested in either generating or purchasing new aptamers to test immunoglobulins against SARS-CoV-2, just in case, let's say, that virus isn't actually transmitted to the human milk, but it's a good way to testing immunoglobulins, or the protective proteins that protect the baby against infection. Test, obviously, against those viruses that do cross and can cause infection in the baby. So like we had said, if the viral load is low, we can confirm that. Bedside test, it actually takes 2 minutes. It's very fast, highly sensitive, like I had said before, and then kind of see whether, if this mother's viral load is not detectable using the GFET biosensor, then, she'll know that immediately, and she could just breastfeed, in that confidence without having to go into a lab or anything like that. This device can hopefully one day be in the home or commercially available. And then what we had already submitted a grant for is to collect human milk from breastfeeding mothers with infants that were admitted to Children's Hospital with a positive viral respiratory panel to analyze and quantify their cytokines, antibodies, as well as viral particles, just to kind of get a sense of what those patterns look like and compare them to clinical severity of the illness. So we'll see whether we get funded to do that, because that would be an amazing project to do. And then another thing is to expand the use of the GFET biosensor into the NICUs to test for CMV in samples of human milk that are given to preterm babies. As I had said before, CMV will negatively affect or impact preterm babies more so than term babies. And we never know if the human milk has CMV or not. And that's it. So I just want to thank my lab and all the collaborators and all the funding mechanisms that help with this project. And I'm happy to take any questions.
All right, excellent. Thanks, Sandra. Do we have any questions?
Sandra, I think you had some data on HIV, is that correct?
No, I didn't include it here. I know we
Yeah,
have the
yeah.
aptamer for HIV, but we haven't run it yet.
I thought there was some preliminary data, but not good enough data that it was chips were not working properly. Is that correct?
Probably.
Yeah. So you do have some data on HIV, but it's not confirmatory because we ran out of personnel. And the
Yes.
chips, since we're relying on chips from China manufactured, that's the main issue right now. And so hopefully in next 4 or 5 months, we'll have the facility to make it in US. It's expensive. That's pretty expensive. That's $30-40 million. But hopefully we'll have something in next 6 months time to make it in US. And then, hopefully we'll get answers to most of them. But the main thing that there's no personnel. Like if you're ready to go for NCIU now, but I don't think you have somebody working with you. Is that correct? So, so that's something that everybody has to think about, how important part is. Now that you had IF and gamma, and you have IL-6 was not very clear. Is that correct?
We have IL-6. We tried another aptamer, and that one worked better.
Worked better? Okay.
Yeah.
Okay. So that you can add it in your next talk. Talk was pretty good, actually. I'm impressed. The only thing you did know, you just a little bit have expanded on graphene transistor. It's just a transistor. The transistor by definition is that resistance is controlled one or other. And here the resistance is controlled by your aptamer binding to your analyte. So that's the gate. Otherwise, it's less like a battery. You apply, put current from positive to negative, like the light bulbs. It will turn on light on. But if you put a blocker in between, which can control, like most of the light bulb that you get nowadays, you can change it, brightness. That's the gate part. So gate is where here is your analyte binding to your probe. And P-base is just a linker that links your probe to graphene. So the graphene sensitivity is single electron transfer. So aptamer is that you have is just 20 nucleic acids. You can go up to 50 nucleic acids. That's our original papers were on PNS for next generation sequencing or methylation and everything. So what it means is that, and it's a conformation based. So that's why it's much more specific than even monoclonal antibody. By definition, even monoclonal antibodies, we have 2 fragments, Fabs. Each one of them can bind. Here it binds to literally 1 molecule. And since it's conformation based, you can use the same aptamer multiple times. Although we don't allow somebody to do it, because why do you want to take a risk where sensitivity will keep going down anyway? But we have done dose response curve to show that yes, if you have to use it 5-6 times, you can use the same aptamer, because it can be reusable. Antibodies are not, because they are covalently bound. Once you bond, it takes a lot of time to get it out. So the electronic, this is truly electrical sensing. There is no electrochemical, nothing else. And the one thing that you missed out of telling that that PNS paper on COVID-19, that was in actually test done in 2 places, in Chicago and in Las Vegas. Once when it was Omicron was very active, once it was first one was active. And each one of them, due to RT-PCR, you need minimum 700 viruses. Here you can test as few as 4 to 5 viruses. So when you're talking about that, in mother's milk, you saw that HIV, if it's negative, question is how negative is negative? What's the dose of HIV that will be transmissible? Can you look at it before? As you mentioned that you can do it in 2 minutes time. Yes, all of the cytokines and all the viruses, you can do in 2-3 minutes time. And next generation system that we'll have, hopefully you should be able to do 100 by 100 tests simultaneously. But again, those things are all depends on where the money comes from. It's a manpower based resource limited system. So So if you want to look at the milk, every panel, everything you want to do, you can do it in a half an hour time. But it does need some support. But right now, for example, you don't have anybody for NCIU to work on it. But system is ready, right? You know now, this grant was the blessing in disguise or God sent grant. There's no question about that. But it was too little for too ambitious project. So, but the point is that,
it was a good start.
Good start. There is no question about that. That's what I'm saying. It was too ambitious project because we had no idea what we are looking for in milk anyway. And two, that the milk that we should have taken, we should have known whether the milk is infected, not infected. Because remember, we have spent 6 months' time in figuring out first. And then we realize that we don't even know what's the milk status is. So as you mentioned several times, that if you know somebody's pre-infected, take the milk, then you get his infected one, take the milk, and then look at it. That will be the best thing to do.
Yeah.
But that you can do it in NCIU system where it's controlled environment, or or in a clinic where everybody goes at 2-3 weeks. Mothers, in fact, I just came back from giving the blood of my wife for testing. I see that there are pregnant women giving their bloods for testing too. So I was going to tell them something to do, but this is not my forte. I'm not MD, so I can't do much. But that's something that you can do. So the prospects of this technology is that you can do at home. You can do it for poor people, rich people, anybody can do. It's easily so. But yes, it does need support.
Thanks, Ratnesh. And I think you're going to give the talk next year. So I wanted to leave some information for you to discuss your amazing creation. So thank you.
Yes, but question, we are open for questions. Remember, one thing that Sandra did not tell you, that we're open for questions anytime, any way, by email, by phone, by by Zoom, whatever you have. Because our job is to make sure that our effort is not wasted, and all the support that you have and trust that you have given to us, that's not wasted. That's the most important part. Is that correct? Is that correct, Sandra? That's what you're looking for? Okay.
That's how we're submitting grants. Fingers crossed.
Right. Yeah. Thanks. Do you have a chat? There is somebody has written you chat. Look into what this says. Okay.
It's just for the survey.
Yes, it would be very helpful if people would fill out the survey. That helps us plan out future seminars and reach out to those interested in giving seminars as well.
All right. Thank you so much. We're out of time. Way over time. Thanks for the presentation. No, it was okay.